The genetic code is universal and is the same in all organisms. This means DNA from a jellyfish can be transferred to a rabbit, for example. However, with all the billions of base pairs within DNA, mistakes can occur. These mistakes are mutations. Often changes in DNA can be repaired by enzymes, but sometimes they persist through generations. Here we see a table showing the genetic code. The genetic code is made up of triplets of bases, sequences of 3 bases transcribed from the DNA molecule to the messenger RNA molecule. The bases are adenine, guanine, uracil, and cytosine. The codons code for an amino acid, such as ACU for threonine. And the order of amino acids determines what protein will be made. This is the process of translation that is part of the central dogma. But when mistakes in replication and transcription are made, the bases in the codon will be different, and thus the protein that is made from the codon will be different. Mutations are changes in the normal base sequence of a DNA molecule. So here's an analogy of the genetic code using 3-letter words to stand for codons. Each letter represents a base, and each word represents a codon. The sentence, "The cat ate the rat," stands for a protein. Let's look at a change in one of the letters of the second codon. We know in English that the letter C and the letter K can have the same sound. So when we read the sentence, the sound and sense of it have not changed. This is an example of a silent mutation because of a different base. In this analogy, the K replacing the C does not change the sense of the sentence, and the protein remains the same. But what if we changed a base, that is one of the letters in a word, to a different one? If we change the C in cat to an H, we have the sentence, "The hat ate the rat," which changes the meaning of the sentence. This is a substitution mutation. We have substituted one letter, which resulted in a very different sentence, and thereby changed the protein the sentence stands for. Now, what happens if we delete a letter? The change is more profound, as every single 3-letter word after the deletion is changed, and the resulting protein or sentence cannot be read. Finally, what would happen if we inserted a letter? The result is similar to the deletion example because every codon after the insertion is changed, and thus the sentence does not make sense. Silent mutations and substitutions are also called point mutations because they change the nucleotide sequence only at one point. Insertions and deletions are frame shift mutations because they shift every codon that follows the mutation. Here we see 2 photos of human blood. On the left we see normal red cells, and on the right we see sickled cells, characteristic of persons who have at least one sickle-celled allele. The sickle cell allele is due to a point mutation. So let's look at the difference in the DNA code between the normal hemoglobin and sickled cell hemoglobin. Recall C stands for cytosine, T stands for thymine, and A stands for adenine. Part of the normal hemoglobin code in DNA contains the bases CTC. But in the mutated sickle cell hemoglobin code, the thymine has been replaced by adenine. So we have CAC. The RNA codons are transcribed from the DNA molecule. In normal hemoglobin, the RNA codon, which is complementary to the DNA, is GAG. But the RNA codon complementary in the sickle cell hemoglobin is GUG. GAG codes for glutamic acid, abbreviated here as GLU. However, in the sickle cell allele, the codon is GUG, which codes for valine, abbreviated here as VAL. This one-point mutation changes the shape of the hemoglobin molecule and causes it to form stiff rods under certain conditions. The formation of these stiff rods causes the symptoms of sickle cell trait in persons who have one mutated allele and sickle cell anemia in persons with two mutated alleles. Sickle cell is one example of a mutation that has changed the evolution of mankind.